Okay, so basically, I think there was only 2 students that said they want to come in earlier for the final exam at 8 a.m. instead of 10 a.m. I did talk to my chair as well as whoever puts together the schedule. I'm going to move it to 10. But those 2 students, reach out to me. I will come in earlier, I will come in later, I will make it work for you guys. You can take the exam, whoever that is. You don't have to reveal yourselves, but come see me. And we'll make a plan for you to take the exam another time, either that day, hopefully that day. I'd like to do it that day, the same day, but or another time. But I think it's just for everybody's purpose. I mean, I see how many people show up late for regular exams at 9:30. To do it at 8, I'm a little worried. So let's, we're going to do it as 10. I'll put out an announcement and make sure it's official. And my chair has to sign off, so everything it's happening today. I'll let you know later. Okay, so let's kind of put that exam, we're going to put it behind us for right now, but, definitely do not, forget to review the exam and make sure that you're using it to study for your final exam. Okay, so I don't know what to do. I can't get the front screen working. I'm going to have to hire these guys to stand here the entire time with me. But we're going to jump into our next unit, which is the nervous system. The nervous system covers 5 chapters. We're not going to do some of them. We're going to only cover parts of 4 chapters for this unit 3, this 3rd exam, covering the nervous system. And nervous tissue was our last type of tissue that we haven't really talked in detail about yet. Even though with our discussion of the nervous system, the muscular system, and the muscle, the neuromuscular junction and neurotransmitter, we actually have delved into a good deal of this material, so it shouldn't be completely new to you. But let's set the stage a little bit for what we're going to be talking about over the next several weeks and the next several chapters. Here's kind of an overview, kind of organizational chart of the nervous system, and we have divisions and different parts, and we can classify it by anatomically, like your central nervous system is your brain and your spinal cord. Okay, the brain and spinal cord are your central nervous system. That's going to be, we're going to cover that in chapter 12. Yeah. And then we have our peripheral nervous system, which is everything else. So we're going to see there are nerves that leave right from your brain. See these that go to your face and things in your head? Those are called cranial nerves. That's part of the peripheral nervous system because they leave the brain or come in and out of the brain. And then all the nerves that leave the spinal cord that go out to all your body, that's also spinal nerves. Those are part of the peripheral nervous system, chapter 13. Okay? Then within, it also kind of includes this, but nerves that go out from the central nervous system, from the brain or spinal cord, and carry signals out. Let's say like a signal that goes to a muscle. We call those somatic motor neurons, right? That would be motor or efferent. So this is more of a functional classification. Signals that go out to an effector, like a muscle, are motor or efferent, and signals that come in from receptors, like touch receptors, pain receptors, anything. Even these little chemical receptors that tell us we have hypercalcemia or something like that. Bringing information in, sensory or afferent, okay? And within both of these, actually, sensory and motor, we have, let's look at motor first. We've already talked about somatic motor. Put a little circle, skeletal muscles. That's what we talked about in chapter 9. Though that was that division, the signals that go out to your voluntary muscles to contract. Somatic motor fibers. And then the motor fibers that go out to, let's say, your liver, your kidneys, your heart, your lungs. Visceral, we call that visceral motor. It's also known as your autonomic nervous system, chapter 14. That's going to be chapter 14. Your autonomic nervous system is for all involuntary motor functions. We don't consciously, tell our blood vessels to get bigger or smaller and heart to speed up or slow down. That's all autonomic. Visceral motor is another word for autonomic nervous system. Sure. Cardiac muscle, smooth muscle, and glands, if you want to circle that. That's visceral. Skeletal muscle, voluntary. So you can write that too. Yeah, there it is. Voluntary, involuntary, skeletal muscle, voluntary, involuntary, cardiac, smooth, and glands, organs. The diaphragm's a good example of a muscle. It's skeletal muscle. It is. Because you can control your diaphragm when you want. You want to take a deep breath, you take a deep breath. You want to breathe faster, so you can. Thankfully, we have a part of our brain that has autonomic signals to our diaphragm. It takes care of those rhythmic sort of things like breathing. Okay. Kind of like blinking also. We talked about some of these examples. Those would still be somatic motor. Still be somatic. Okay, and then we're going to get to this. Don't worry about it so much right now. These two divisions of the autonomic nervous system. We have these two competing divisions, the sympathetic nervous system and the parasympathetic nervous system. And the best way, we always say sympathetic. Does everybody know the little phrase you always hear with sympathetic nervous system? Fight or flight, you ever hear that? Yeah. Your fight or flight. It's like when your body's getting ready for activity, and we think of extremes, like when you get really scared or really you gotta fight or you gotta run or you're, threatened. All those things. Your heart rate starts increasing, breathing increases, your pupils dilate, the blood goes to your muscles instead of to your internal digestive organs, things like that. Yeah. It's part of the autonomic nervous system. It's one of the divisions. Fight or flight is here. And then this would be the other one. We call this, we'll see chapter 14. Don't worry about it right now. But I call this one resting and digesting. It's like when you're slowing down, when you're repairing after activity, when you're sleeping, when you're digesting food, when you're, doing like body maintenance, repairing stuff after you've damaged it. Okay, so those are sort of there's a lot of terms in here: sensory, motor, somatic, visceral. I think it's all explained in the next slide here. Yeah, so you have a little description of everything there. There it is: fight or flight, resting and digesting. But the visceral motor, autonomic nervous system, somatic motor, skeletal muscle. It just refers to the diagram before it, so you can review that. What happened? Why does it keep doing the same screen? Okay, that's weird. All right, so by the way, I did update these notes. If you printed them out before last night when I uploaded them, they're going to be similar, but there's a new version. I put a couple different things in a little bit of a different order and maybe messed around with the fonts and that kind of stuff. Nothing major. Okay, so with the nervous system as this type of tissue that is kind of unique, we have two types of cells. We got the main cells we're going to spend most of the time talking about. Those are your neurons. Those are the nerve cells. These are the cells that carry that we say they initiate and propagate or spread these action potentials, these electrical impulses, signals. And then we have these support cells that they are not electrical cells. They are not, these responsive cells to stimuli. They just support the neurons. They protect them in various ways we're going to talk about. Okay, we call them glial cells for short. That's commonly used, but neuroglia or neuroglia, neuroglial cells, they are support cells. They're smaller. They're not these long. Neurons can be very, very long. These are usually much shorter. And some of these cells, actually, you can see them in green, they wrap around the neurons. The neurons are in yellow there. They insulate them, kind of like a wire. You have the plastic on an outside of a wire. Okay, but we'll go through some of them. I don't know why it's doing that. Yeah. No, glial cells have nothing to do with they are not electrical cells like neurons are. Okay, so here's our fourth tissue class, nerve tissue, and the neurons are really the cells that give nervous tissue, its function. And they are excitable, so even though that's not unique yet, because we know that muscle cells are also excitable, okay, they conduct. So we can, one part of the cell gets stimulated, just like a muscle cell. We stimulate them at the neuromuscular junction, then the rest of the muscle cell becomes, it propagates or conducts this action potential all the way along the cell. Nerve cells do the same thing. But when muscle cells, we said were unique because they contract in response to that stimulus. Nerve cells, all they do in response to stimulus is, they spread it to the very end, and then it releases a chemical. It secretes a neurotransmitter. Okay, so they secrete a neurotransmitter onto another neuron, onto a muscle, onto a gland, onto something to stimulate some effector. Okay, so that's the characteristics or properties of neurons. Okay, let's talk about the parts of a neuron. I think this is the part of the notes I switched around. I did glial cells first in the old version, then I did the neurons. I did it backwards this way. Okay, so a neuron, we have the three major important parts to know of a neuron are the cell body right here, which is called the soma. Okay, and let me just see, I think I put the next slide with the three parts. Yeah, okay, so the soma, you can see one large nucleus in the middle. The nucleolus, remember the nucleolus is the part of the nucleus where we make what? Anybody remember? What do we make? What's made in the nucleolus? Ribosomes, I heard someone say it. Who was it? Take some credit. Okay, ribosomes, and you're gonna see, we'll see in a second, you see lots of ribosomes on the outside and this rough ER because they make a lot of proteins, a lot of neurotransmitters and things like that. So let me come back to that in a minute, but let's just go here. So the soma is this whole thing here, the whole soma, that is this, or cell body, but soma is an easy way, we use that term a lot. Attached to the soma, you'll have multiple branches. Sometimes you could have a neuron like this, where the cell body is like this, and then it has a process, and all the dendrites are here. These processes, they're not coming directly off the body, but this is very common. These are dendrites. These are receptive regions. We could have, let's say, another neuron from far away comes all the way here and synapses with that. Every one of these little endings can have a synapse. So these receive signals from other neurons or from stimuli. Sometimes, maybe this is like a touch receptor. You touch that and it sends a signal. So dendrites are the receptive region of a neuron. What was I just going to say? Shoot. Anyway, it'll come back to me.
They only receive signal from other neurons?
From other neurons, but there are some neurons have specialized dendrites that are nerve endings, like in your retina of your eye. The specialized dendrites respond to light waves hitting them. You can have dendrites in the tip of your finger as part of a neuron that actually responds to pressure that starts an action potential, or from chemicals like smell and vibration in your ears, special senses like that. Okay, so we have the soma, we have dendrites, and then we have one long process coming off of the soma called the axon. Okay, and signals only travel this way. Okay, they go from the soma this way down to the very end. And we have at the end of the axon, we have axon terminals or synaptic knobs, right? Those are going to be the things that we saw attaching to a muscle, but they can also attach to the dendrite of another neuron. Okay, we can have these circuits or these sequences of pathways where one neuron stimulates another, which stimulates another, which stimulates another. I just want to make sure I don't forget to do this. I don't know if you're here, but someone left their book class. Basola, are you here? No? Oh, she is? All right, if she doesn't come, then you can make sure she gets it. Okay, so we're going to get to these Schwann cells. This is an example of a glial cell that wraps itself around the axon, and it insulates and protects it, but we'll get to that in a minute. Okay, so dendrites, axon, soma. Those are the three major parts, and I kind of did that here. So the soma, you have one nucleus. The nucleolus, you can write, makes ribosomes. And there's a lot of rough ER in ribosomes, and they use a stain. It's called Nissl stain. And it helps when you're looking at tissues, they use this stain to see where the nerves are, because it really sticks to that endoplasmic reticulum and the ribosomes. And it gives it almost like a leopard print, they say. It gives like the cell this like leopard-looking appearance, spots. So they're called Nissl bodies. It's referred to a lot in a lot of the images in the chapter, so I wanted to just put it there, but I'm probably not gonna put that on the test, but just so you know what that is. The dendrites, again, receptive regions of a neuron. They're directly branching off of the soma or at the end of a process like I just drew. We're gonna go over a couple different shapes of neurons that are different. And then the axon is this long, slender process. Some of them can be extremely long, almost the length of your entire body. That conducts action potentials, remember APs, we're going to be talking about APs a lot today, away from the soma. Okay, so it always goes signals, here's the soma of a neuron, there's its axon, okay, maybe some receptive dendrites. Okay, signals go this way. They come from the dendrites to the soma and then the axon. That's the direction of the action potential or the impulse. Okay, a couple things. Oh, really important spot. It's on the picture here. But this area, let me just highlight this. This area right here, super duper important. We call it the axon hillock, but it's also known as, and I like this name better, the trigger zone. Trigger zone. They also call it the initial segment sometimes, like the first part of the axon, like right where the body, the soma is becoming the axon. This is where action potentials start. If a neuron, I'm going ahead a little bit, but if a neuron gets enough stimulation from other neurons, enough to reach a certain voltage, it's going to generate an action potential. Remember that little spike we did on the graph? It's going to start there, and then it'll be another one, then another one, then another one, and then it goes all the way down to the end and makes it release a neurotransmitter onto the next neuron or a muscle or something. Okay, so the axon hillock trigger zone is the first segment of the axon as it leaves the soma. This is where action potentials originate. We're going to talk a lot about that in a bit. At the very end, we said we have the axon terminals or synaptic knobs, which have synaptic vesicles with neurotransmitters in them. And there's lots of different types of neurotransmitters. We've only talked about acetylcholine, pretty common one, but there's lots of other ones. They think there's almost like up to 50 different types of neurotransmitters, but we're only going to cover a couple of them.
So when the action potential leaves the axon and just goes over the dendrite, let's say, the action potential just like ceases to be, or
When it leaves,
like the axon, let's say there's like a chain of axon and then dendrite.
dendrite, yeah.
So then once the action potential gets to the dendrite, it just kind of like
Well, let's say we have a neuron coming from here, it comes here and it releases a neurotransmitter onto this dendrite, right? There's a synapse. It's going to stimulate this a little bit. If there's enough stimulation, maybe several of these are stimulated at once, or maybe a bunch of signals come really fast. If there's enough to reach what we call a threshold voltage, we'll generate another action potential here and it'll go to the end of that. And then eventually it's going to get somewhere. If it's going out, maybe it gets to a muscle, makes the muscle contract. Or if it's coming into your brain, it goes to a part of your brain and it triggers, it stimulates part of your brain. I don't know if I answered your question. Sort of. Okay. Maybe hang in there. I think it'll become clear. And if not, ask me again. Okay. So just some terms like the axon, the cell membrane of the axon. Remember the sarcolemma of a muscle? We call it the axolemma of an axon. Because the electrical signal, remember we said the electrical impulse travels along the surface of the muscle cell, right across the membrane. It's just sodium going in, potassium out, sodium in, potassium out. Like that. Same thing on the axolemma. It's the plasma membrane of an axon. And the cytoplasm, not sarcoplasm, we call it axoplasm. Axoplasm.
and they all have like you said that the action can be this is still like happening.
Yeah, we're going to talk about some of the speeds just to give you an idea. But some of these neurons can conduct signals at like 120 meters per second. I mean, that's like lightning, Even some of the slower neurons are still, you know, we're measuring them in meters per second, 5 meters per second. Think about that. That's pretty quick. I'm going to have a slide on that in just a couple minutes. All right, so just some of the terminology, know the parts of a neuron. There's a pretty, you know, you might want to put a little star in this diagram, know the parts of a neuron. There's the description. I don't know what's happening. Okay, so now let's talk a little bit about these glial cells. We're just going to kind of go through them and label them here. We'll highlight a few of them here as it pertains to, you know, just nerve physiology. But when we get to the brain, we're going to see like all of these in the top box. These are found in the brain and mainly the brain, the central nervous system. And these two, and it's mainly just this one we're going to talk about, found in the peripheral nervous system. And this is just, you know, so we don't know a lot going on here yet. Well, I have a slide for each of these. But for example, like when we get to the brain, we're going to talk a lot more about astrocytes and epidermal cells. We're just kind of listing them here, but you'll see in the next chapter we're going to talk about them more. But these are examples of glial cells or neuroglial cells that do not conduct signals, but they have other roles. So for example, here is a type, this is just kind of a picture of a bunch of them, but an oligodendrocyte is a cell, look at them here. They reach out part of they grow these little extensions and wrap their cell membrane around an axon. The axons are in yellow. So these are like a bunch of neurons in your brain, you know, maybe going one place. They insulate them, they wrap around them. They have processes that form, we call this the myelin sheath, okay? This is when these glial cells, there's a different one in the peripheral nervous system, they wrap around the axon of the neuron. Think of it like the axon of the neuron is like the copper in a wire. It's the metal inside a wire. But you know, you just don't have bare wires in your wall. That's a hazard. You have insulation around it. There's the different, you know, red wires, blue, green. That little waxy plastic covering, oops, sorry, are, you know, basically the same kind of thing. It insulates it. It keeps the signal, you know, in one neuron. It keeps it from like jumping from this neuron to this one, right? Because this could be going a different place. And it also protects them a little bit. It can actually hold several of them together. Maybe this is in the central nervous system, we're going to see in your brain, you don't have nerves in your brain or your spinal cord. You just have neurons going this way and that way. In the peripheral nervous system, you have nerves. You have connective tissue around these neurons. You know, there could be 1,000 neurons and it's in a tube. You could see it. So this kind of holds them together, kind of, you know, keeps things connected, and it forms this myelin sheath. So the myelin sheath is the insulation around an axon. One interesting thing to notice is that we can use this analogy of you know, neurons being like wires in your home, you know, carrying electricity. And the analogy holds up for the most part, but in certain cases, there's differences, obviously. I mean, look at these little gaps. You have these little gaps between these myelinated sections. We're going to talk a lot about them when we get to, you know, talking about the propagation of action potentials. If you saw a wire like that on your wall, you would have to replace it. That is a fire hazard, okay? So, you know, they should be continuous. If you make a nick in the insulation of a wire, that could cause a spark and that could be a problem. But these are going to be really important. We're also going to see that a myelinated neuron, when it has myelin, because not all of your neurons have myelin on them, but the ones that do conduct a lot faster than ones that don't. It allows the signal to travel a lot faster. I have a slide on that in a bit. We'll talk about the speeds of nerve impulses. Okay, but here, these oligodendrocytes, and by the way, because we say that neurons, I don't know if I put this, how about I ask it this way? Neurons do not have centrioles. What does that mean that neurons aren't able to do? Mitosis. They do not divide. Neurons do not divide. Yeah, they're amitotic. They do not divide.
They don't regenerate, right?
They do not. I mean, there are certain areas of the brain where neurons can regenerate or very slowly regenerate. Oh yeah, I had a hernia operation, I mean, when I was in college. And I remember after it was done, like in my thigh right here, I mean, they do it like here, inguinal. It was an inguinal hernia, but a little low. I had this big patch of numbness on my thigh afterwards. And like, years, it's still a little numb, but it was much bigger. It got smaller and smaller. So there definitely was some nerve repair there, but sometimes, yeah, like a surgery like that, it could be cut and the nerve doesn't grow back. Okay, so, what I was getting at is that because neurons, right, neurons are amitotic, when you hear someone has, like, God forbid, a brain tumor or something, they might have a name like an oligodendrocytoma or an astrocytoma, because the glial cells can go through mitosis and form tumors. So a tumor in the brain isn't made of neurons, it's made of glial cells usually. Okay?
Question.
Yeah.
You say there are no nerves in the brain. What's the difference between nerves and neurons?
I didn't hear the last part. What is?
Difference between nerves and neurons.
Oh, good question. Okay, so a nerve is like the difference between a muscle and a muscle cell. Okay, so you could have a nerve like this, like you do a dissection, you look in the arm, you see this long nerve, it's kind of like, let's say you draw like a tubular nerve like that, it's a nerve. But a nerve can be filled with thousands and thousands of neurons, okay, inside a nerve. It's like a big cable, if you cut a big cable and it has like, 100 wires in it. Those wires are the neurons, but the big cable is the nerve. Okay. Like we can have a somatic motor nerve to a muscle. But remember we said motor units? We can maybe only let's say it's a nerve and it has 10 neurons in it. It's going to be a lot more than that, but let's just say there's 10. I can only be firing 5 of those for a light contraction, but I can fire all 10 of them or all 1,000 of them, to get more. So that's the difference. All right, the next one are called astrocytes. These are the most abundant type of glial cell in the brain. And they kind of fill in a lot of the open space between neurons. Look at them. They have lots of processes also. They wrap around neurons, not so much to insulate them, but to keep them connected to one another. And also, they do a very important thing. Look at this capillary, blood vessel. They wrap around the capillaries. And what they do is they form part of what is called, let's write this in here in your notes. BBB means the blood-brain barrier. We're going to talk a lot about this in the central nervous system chapter about the blood-brain barrier. But it's really important that we just don't let whatever happens to be floating around in our blood get into our brain. There's extra security. We have a bunch of extra security in our brain. So not only are the capillaries that go into your brain have a lot more tight junctions between the endothelium, remember the little simple squamous in there? They're really tightly connected because it's like, you could ingest like a toxin or something and you're like, okay, we're okay if that damages part of your body down here, but we got to keep this guy safe up here. So not only the capillaries have these really tight junctions, we have these astrocytes wrapping around them, and anything that wants to get from inside here to, out has to go through not only the wall of the capillary, but through these as well to get into the brain tissue, into the fluid, the extracellular fluid in the brain. Okay, so it's protective, it protects the neurons. As a matter of fact, if neurons die, which can happen if a neuron loses its blood supply from like a stroke or trauma or lack of oxygen, whatever, the neurons die, they don't come back. And the space will be filled in with astrocytes. They fill in the space, and they call it a plaque. And you can see it on like an MRI. Like, ooh, after a stroke, you might have this area of your brain. It's not functional anymore, but it's just this scar tissue. So it forms this, we call this sclerotic tissue or scar tissue made of astrocytes. But you might hear that term, you might notice it now. They talk about astrocytomas. It's a tumor of the astrocytes that form this mass. Tumors in the brain, even though, they're not neurons, these tumors, hopefully, God forbid you ever get a brain tumor, but hopefully if you do, it causes symptoms early. let me give you an example. Like if a tumor gets big and it compresses, let's say, a bunch of neurons, or it compresses a blood vessel going to some, and those neurons are for vision or for something else, or they cause pain. You would feel that, you go get checked out, and hopefully the tumor's small enough they can treat it, get rid of it. Yeah, but sometimes these tumors can go quite large before they press against something that you notice. And at that point, sometimes it's too big or hard to get to. My brother-in-law had a kind of tumor that was developing, and I think I mentioned this. He was just talking about headaches, and it felt like a migraine that he used to get when he was younger. And he just, said he reached out to get this medication, but he just mentioned something. He's like, I'm just getting this weird visual thing, like I can't see in my peripheral vision. And he was telling me and my brother, and we were like, okay, you should go get checked out. And basically, the neurologist in the hospital said if you would have waited another hour or so, you might have been gone, because this thing was ready to explode like a blood vessel in there. So it caused some weird, symptoms like that. So you don't want to mess around with that stuff with the brain. If you're getting visual disturbances, or you're getting, you don't want to be a hypochondriac and stuff. You get those little floaters in your eyelid. We get people like, oh my God, I'm having a stroke. But something noticeably different, and even things like a change in your sense of smell and with other senses, because that could be a sign of something going on that is, they call it the mass effect. It's not the neurons themselves it's the pressure being put on them by a tumor that's growing, pressing against this other normal tissue. Okay, so astrocytes are the most abundant. They fill in a lot of the empty space, protecting neurons, insulating them a little bit. Not so much like they're not causing like blood flow, they're not contractile, they're just an extra layer of protection. It's like this blood-brain barrier. So it's like an extra level of security for stuff that gets through. And we also have, I'm going to skip over for a second, we also have, let's say here's a blood vessel, okay? We have these tight right? Simple squamous. We got these tight junctions. It doesn't show the astrocyte here around it, but just picture that, they could be around it. Let's say like, astrocytes are forming this other layer of defense. We also have microglia, which are immune cells. They're macrophages. They're like the next line of defense. Something does happen to get through. Maybe does it say a microbe or so? Yeah, invaders or microbes. Maybe they happen to get in there. These microglia, just like in the skin, remember we had the dendritic cells or Langerhans cells? In all of our organs, we'll have cells that sit there, and in case something does get through, they identify foreign substances, gobble them up, they swallow them up and clean up any debris. I say surveillance, looking for any microbes or invaders, any viruses, stuff like that happen to get through. And when we learn about the immune system next semester, you'll see these cells are really important. We call them macrophages, or we give them different names in different tissues. We call them microglia here. But they can alert the immune system. So not only do they clean up and gobble up, maybe they gobble up a little bacteria or something that happened to get in there. They're not just going to clean it up, they're going to bring it and show it to these other immune cells, and we're going to make an army of cells that are trained to find that exact type of bacteria all around the body, and then we make millions and millions of them so that we can fight off an infection. Anyway, microglia, they're kind of part of our immune system. They activate the immune system, but they're cleaning up dead cells, any microbes, foreign substances, cells that died. We clean them up. Okay, ependymal cells, I'm just going to mention here. Let's not worry about them right now because we're going to get to them. But when we learn about the brain, we have these areas in our brain where we produce a fluid called cerebrospinal fluid. Yeah. Yeah. They connect, they keep neurons connected to one another, but they also surround blood vessels to form part of the blood-brain barrier. Okay, so we have these fluid-filled cavities in the brain that contain cerebrospinal fluid, CSF. And we're going to learn about this in the next chapter and why CSF is important. But it is, we would have like, let's say we have like blood vessels in the brain here. you know, and we're gonna have our protection, we'll have our cells, we'll have our astrocytes, but stuff, it leaves here, and to get into the CSF, it has to go through these ependymal cells and go into the CSF. So for now, let's just keep it real simple. Let's just say ependymal cells help produce the cerebrospinal fluid. Okay, I think that's fine enough. They control the composition of the CSF. Let's just say they help produce cerebrospinal fluid. Just put that in the back of your head. We're going to fill in a lot more detail on that when we get to this chapter. But it's a type of cell found in the brain. It's obviously not a nerve cell. And it's kind of like it's like almost like a cuboidal cell with some cilia. And it just forms the barrier of these what we call ventricles, or CSF-filled cavities. Don't worry about so much now. Yeah.
This is all part of the glial cells, right?
These are all types of glial cells. So those are the 4 glial cells you'll find in the brain: oligodendrocytes, astrocytes, ependymal, and microglia, or microglia. And then in the peripheral nervous system, picture a nerve going through your leg or your arms or somewhere in here. And, here's another shape. Here's a neuron with the somas up here and then the axons down there. But we have these Schwann cells, which myelinate, just like oligodendrocytes in the central nervous system. Schwann cells are in the peripheral nervous system, and they actually, it would be a cell like this, like there's its nucleus. It wraps itself around. Let me show you in the next slide real quick. Oh my goodness. Okay, see how a Schwann cell wraps around the axon, then wraps and wraps, and it can wrap around hundreds of times. And the cell itself wraps around and insulates a little section of it. Then in a cell next door, another one, another one, another. Okay, so Schwann cells myelinate neurons in the peripheral nervous system. I'll go over some more detail in a second. And then we have this other type of cell. We're learning more and more about it, like what they do. They're just called satellite cells. I usually don't even test about these, but I guess just know they're a glial cell. And they protect the soma. They're just sort of around it. They may also help in, like, let's say, there's, maybe neurotransmitter that is used up or broken down. It could reabsorb it back in here so that, but let's just say it helps protect the cell, protects the soma, helps provide nutrients and things to the soma. Not so important. That's probably the last time I'm going to mention satellite cells in our whole discussion.
Why does the one cells represent
Well, they just form this, insulating barrier. And like I said, a neuron that is myelinated like this is going to be able to conduct a lot faster. Okay. So here is the Schwann cells form this myelin sheath, just like the oligodendrocytes. So let's talk about this myelin sheath. It's actually the cell membrane of these glial cells. So it's mostly lipids, right? The cell membrane is mostly phospholipids with some proteins in it. So same thing, mostly lipids, and it has a whitish color. It has this whitish color, and when you ever hear like white matter and gray matter of your brain? You may have heard that. White matter is myelinated neurons. White matter is myelinated neurons. Okay, so it has a whitish color, and it allows neurons to rapidly conduct action potentials. Okay, so in the peripheral nervous system, we think of Schwann cells. They wrap themselves around. And we said in the central nervous system, it's oligodendrocytes, which it's thinner. The myelin sheath is thicker in the peripheral nervous system because it's exposed to more potential damage. Your brain is inside your skull. There's no muscles contracting in there. There's no moving parts in your brain, right? Whereas in your arm, you have muscles and things, and in your body. So it's more protected. Maybe that answers your question, why it's wrapped so many times there. And oligodendrocytes, one oligodendrocyte, as we saw in that picture before, can wrap around several neurons and kind of hold them together. Schwann cells just wrap themselves around the axon. Okay? Now I want you to notice these little spaces between the segments. We saw those before. They are called nodes of Ranvier. We can even put a little Ranvier. Okay? Nodes of Ranvier. They're really important. I'm going to just show you, because I think I forgot to put this on a slide, but it probably comes up in another chapter. We also noticed, remember that initial segment, the axon hillock or the trigger zone? That's unmyelinated, that first segment. And then you would see myelination. Let me see if that's in the picture. Not really, but well, like right here would be like the axon hillock, and then it's myelinated. Something like that. But let me show a neuron like this. There are some neurons that picture this without, I'm deciding if I should draw, I'll do it up here. So let's just say we have a regular neuron like this, maybe some dendrites, one of those ones that we saw before, and then an axon like this. And let's say it's unmyelinated. The action potential is going to start here at the trigger zone, spike, and then it's gonna form another spike, another one. It's going to travel all the way down the axon, axal lemma of that axon, all the way down, unmyelinated. Whereas myelinated, let's say the action potential starts here, it's moving down, it jumps, it goes here, and then you see it there, then you see it there, then you see it there. And that type of conduction is a lot faster than if it has to just go, each action potential triggers the next one on the membrane. By jumping from segment to segment, it goes a lot faster. And we call this, I want you to write this down because I don't think I put it anywhere else. This type of jumping from one node to another, these nodes of Ranvier, did I mention that? Did I list that here? Where is it? It was on another slide, right? Well, let's put it here. This is a node of Ranvier. Okay, so it jumps. And that is called saltatory conduction. So write it down. Saltatory conduction is the conduction of an action potential along a myelinated nerve. And I always thought when I first learned this, like saltatory, and I know action potentials, we're going to talk about sodium going in, potassium, like salts. So I'm like, oh, let's do a salt or something. And then there it was with my little daughter watching Dora the Explorer. And she's yelling at us to, get up and, what are we doing? And she's going, we have to jump. And she's going, salta. And I'm like, what does that mean? Oh, saltar. Anybody habla español? Saltar means to jump. I went, oh, saltatory conduction. It's jumping. So Dora helped me remember that. Salta means to jump. And it's jumping from node to node. And it goes a lot faster when that happens. So that's why myelination is important.
So if it's unmyelinated, it doesn't have any.
Right, there's no myelin, so like this, so it would just propagate all the way down without skipping. This like skips, it's taking shortcuts. You'll see why. We'll get to the physiology of it later. What time is it?
That would be.
We got 5 minutes, but we
That
won't
would
get
be
to
saltatory.
it. This would not be. This is non-saltatory conduction. I don't even know if there's another name for it, but just regular conduction. This is saltatory. And when you talk about like, well, why aren't all of our nerves myelinated? Why wouldn't we want just faster conduction along them? Well, myelin, to make myelin requires energy, it's extra mass we got to carry around. If we don't need it, for example, let me see if I have here. Okay, this is just showing the cell wrapping around. I'll come back to that in a second. There it is, cut in half. You can just see. I don't know why. I got to fix that. So like, why would some be? We want fast signals from like, receptors, let's say in our muscles, or like touch or pain receptor. You touch something hot really fast. You want to be able to respond to that. If I'm tipping over, I want to be able to, really fast. So those types of things, skeletal muscles, vision, balance, these are all myelinated fibers very, very quickly. But the signal that says, there's a little, maybe a pressure receptor with a nerve inside my stomach. Hey, food just got here. And then it sends a signal, to somewhere to secrete an enzyme. Does that have to happen, lightning fast? No, it can be a little bit slower. So a lot of nerves like in your digestive system and in things like that, they're going to be unmyelinated. It's just less, so we don't have to use as much energy and materials if you don't need them. But there are certain neurons that we want to have this lightning fast connection, propagation. Let me back up 1 second here. These are just a few images of the myelin sheath. I just want to go over one or two terms here. Let's see. All right, so it wraps around, and then we have, so the outer part of the axon, let's label this. This is the axon, so that's the axolemma. Right, that's the cell membrane of the axon. Axoplasm would be in there. the Schwann cell, the outer layer of the Schwann cell is called the neurolemma. This is the outer layer of Schwann cell. Just gonna write Schwann. I spelled that wrong. S-C-H-W-A-N. All right, that's the neurolemma. If it's myelinated, it has a neurolemma. If it's not, it doesn't. Anyway, not super important. Myelination occurs, a lot of it's happening towards the end of fetal development and in infancy. That's why when you look at, breast milk composition or baby formula, really, really high fat content, okay? Because they're building this myelin sheath. Does anybody know there is a disease where a myelin sheath breaks down? Does anybody know what that's called? Myelin sheaths are destroyed, and that obviously affects the conduction of neurons. It slows it down, it causes errors, maybe the signal jumps from one nerve to another. It's called multiple sclerosis, MS. Multiple sclerosis, MS, is the damaging or breaking down of the myelin sheath. What's that? Oh, it can. I mean, it can cause a lot of muscle weakness. It can cause a lot of, because those myelinated nerves, problems with coordination, muscle coordination, balance, all kinds of stuff. There's lots, it depends which nerves it affects the most, and it can cause symptoms on those nerves. Okay, there again is myelin sheath. Yeah, there you can see the axolemma and the axoplasm, that's the axon, and then the neurolemma would be outside. But anyway, that's enough of that. We just got a second here, let's see. Okay, so just we'll end with this. You don't need to know these numbers, okay? But just look, obviously, there's two factors that affect the speed of conduction. And I'm not gonna go in crazy detail. Your book goes into like type A fibers, type B, type C, and then the subclasses, but based on size. The larger a neuron is, the faster it conducts. The larger a neuron is in diameter, okay, so let's say increased diameter of a neuron, faster. But the main thing is the presence of myelin, okay? Myelin faster. More important. Okay, so a very small unmyelinated fiber, 0.5 to 2 meters per second. A small but myelinated fiber, 3 anywhere up to 75 meters per second. But these large myelinated fibers, like these type C fibers, we call them, go to our muscles and things, and those it can be super fast. Okay, like, dilating your pupil, it gets dark and your pupils get a little larger just to let more light in. Like, that doesn't happen, it's like, okay, a little bit slower. All right, guys, we're out of time for today. I'm not going to put up the quiz for chapter 11. Obviously, we're not done, but the study module, definitely check it out. There's going to be some videos and tutorials, lots of videos also that I put in the folder for chapter 11. So do that. I'll be doing a booster session at noon today if you want to join for that. We can go over the exam. We can review this stuff, whatever you'd like to do, or if there's lab stuff you want to go over, we can do that as well.